Hi, my name is Elna Gottlieb. Welcome to DNA Learning Center Live, DNALC Live, for another session. And so if you did tune into my DNA structure video, this is sort of like the next section. So we're going to talk a lot about RNA transcription today, and you'll learn all about what that is. We're going to also be building a model to kind of help cement it in. We're going to be using some of these again. We're going to be using some of this. Some pipe painters, and so hold on one second. Okay, that just popped on. All right, so now first of all, what we're going to talk about, if you did check out my teaser on Instagram, I had a little 10-second clip. We're going to take a look really quickly at that 10-second script. So I'm going to share my screen right now. Pop on the screen. There we go. It takes just a couple seconds. Okay, and we are going to go to, oh, what's this? Oh, maybe we won't do this. I don't know why this is coming on. Oh, okay. I don't know why I need that. I don't know what's going on with that. All right, so, okay, we'll skip that. What is this? Anyway. I'm going to come back. All right, I'm back. All right, forget about that clip right there. So my thing went out, probably my Wi-Fi or something. But anyway, so now basically I had a little Hot Wheels track, and it took 10 seconds to go through the entire track, 10 seconds. And it took about 4 hours to build that, and then 10 seconds for it to all be over. So now, the processes in your body happen a lot faster than that. In fact, they're measured in nanoseconds. And nanoseconds, those are fractions of seconds. And so to make you, to make all the traits that you see in yourself and other people and all living things, there are things going on in your body that are happening in fractions of a second. And we're going to explore one of the most important processes that develop you and the way you are. And so we're going to look at sort of one of the masters of those processes and one of those like fast things that happen. Now you might be thinking, okay, yeah, so we're going to talk, we're the DNA Learning Center. So we talk a lot about DNA. And so actually it's funny, this is not the master of the process. A very, very important, crucial role in this. So DNA is extremely important in everything that your cells are doing to make you, you. But there's someone who actually, basically kind of talks to DNA and communicates and is involved with DNA quite a lot. And so we're going to talk about that. It's another molecule, very similar molecule, just a little bit different. So for this, I'll share my screen. There we go. For real this time. Not pretend, because this will work. Okay, there we go. All right, so oops. There we go. And so we're going to talk about a molecule called ribonucleic acid. And so ribonucleic acid, there is your R, your N, and your A. So that is RNA for short. And so that's how we usually hear it, RNA. And so I like to think of RNA as sort of like the secretary for DNA. Now, DNA is pretty similar. And so DNA is also an abbreviation for deoxyribonucleic acid. Whoa, it's a mouthful. And so they're only different by a little bit, but DNA does tend to be longer than RNA. So we'll talk about that, but it can be broken down. So let me break this word down. So you have nucleic, that means the nucleus of a cell. And that's the same in DNA. And acid, that's talking about the pH. So that's from acids to bases. And so that means that it tends to be a negative charge. Same thing as DNA, also negatively charged. But the difference is right here, the ribo. So ribo stands for ribose, and this is deoxyribose, right there. And when you see OSE, that tells you it's a sugar. And so that's a sugar right there. And so they're only different by a little bit. Now, when you see DE, ironically, because DNA is much bigger or much longer than RNA, you might think, that DNA has extra parts to it. But actually, the DE in there tells you that it has one less type of atom in there. And so there's one difference between ribose and deoxyribose, the two sugars. If we look at my next slide right over here, and I'll get rid of this little box. And so we have ribose and we have deoxyribose. These are their chemical shapes. And so this is when the chemistry all comes together. These are what the molecules sort of look like if we were able to look at them. So you have your ribose and you have your deoxyribose. And when you compare them, you'll see there's really one difference there. There's one little simple difference. I'll let you look at it for a second and see if you can figure out that difference. It's like one of those games. They have all those games where you have to look at the two pictures and pick out the differences. Now, as you were looking at that, if you noticed that here's a difference right there. And so you are missing an O in that spot, that's the difference. So deoxyribose has one less oxygen in that spot than ribose. And so the DE means like one less or less. And so now, but just like deoxyribose, ribose connects to your sugars and to your phosphates. It is a sugar. So you have your ribose sugar, and it connects to your phosphate. And then, of course, there's another ribose right there. And so on this side, you have your deoxyribose. I'll just use a D for that, connecting to a phosphate, and then connecting to another deoxyribose. So essentially, they're the same thing. Now, because of that, I'm going to come back, stop sharing my screen for a second, and just kind of bring you back to here. There we go. And so now if you think of DNA right here, and so it has these two sides, RNA is a lot simpler. DNA has this spirally shape right here, but RNA is more like a line. And because of its sugars and phosphates, it is twisty also, because that's what gives this twist, the way the sugars and the phosphates connect. So they have this sort of twistiness. And RNA is the same way, but it's a single helix instead of a double helix. And you're going to see why. You see, in order to use the DNA, and really to do anything with this DNA, you can't use it like this. When it's together, it's almost like having the book shut. You need to open the book. And so the way DNA opens, it unzips. So it literally will unzip. The hydrogen bonds will split down the middle. There we go. It'll open up. And so if you're making new DNA, then new little, we call them nucleotides, that's a base, like an A, T, G, or C, will connect to a sugar and a phosphate. They'll come in and they'll reconnect, and they're going to fill in the sides, each of these sides, like that. And eventually this whole strand will be two separate strands. But when the DNA is open, there are other things you can do too. And so what happens is RNA will come in and it's going to read this section right here, and it's going to what we call transcribe the information. Now I'm going to describe that a little bit better. I'm going to go back to sharing my screen. Oops. There we go. Takes a couple of seconds to get back. There we go. Oops. Keep doing that. There we go. Okay. And so now the whole idea of DNA, let's go back and kind of review the idea of DNA. And so you have, we call it the central dogma. I'll just write that. So, because that's something that you see in biology a lot, central dogma. Dogma is just something that's kind of a standard idea that a lot of people accept to be true. Central dogma specifically kind of surrounds the idea of how DNA works in biology. And so you have a cell, you have your cell membrane around the outside, you have your nucleus. Inside that nucleus, you have your DNA. And here they're organized in these little bundles, which we call chromosomes. Now we have 46 chromosomes that we inherit from our parents, but and this is an actual photograph of chromosomes, and so is this right here. So that's a photograph of a chromosome. And you have dark spots and light spots along the whole chromosome. But basically, if I unravel that chromosome, you're going to get to that DNA. Now, on that DNA, if you consider like all of the DNA to be sort of like your cookbook, then your actual recipes are what we call these sections that are referred to as genes. And so those sections, those genes are responsible for producing traits, but there's a lot of steps that go between genes and traits. And so genes are recipes to produce or control or manipulate proteins. And proteins, I like to think of proteins as being sort of like the building blocks of traits. So you go from proteins to traits, whether they control a trait, like how much protein you make will, you'll achieve a certain height if it's human growth hormone and things like that. So basically, your genes are recipes for proteins or to manipulate proteins, and those proteins are making your traits. And so, but there are steps in between there. And to get that information from your genes to create those proteins, well, you don't really want to have that DNA leaving the nucleus. It's nice and safe in there. And so it's like, you want to try to preserve your cookbook, so you keep it in a safe place. And so in this case, we want to take the message and bring it out of the nucleus and bring it to where it needs to be. And that's where RNA comes in. And so RNA is a molecule that's very similar to DNA, but it has a few key differences, other than the fact that it has an extra O, a little extra oxygen on there. There are other key differences. Now, DNA has these base pairs that we're probably pretty familiar with. A for adenine, T for thymine, G for guanine, and C for cytosine. And they pair up to make the base pair. So those are your bases. A loves T, the little hearts, and G loves C. And so when they come together, we call them base pairs. And so there's two types of base pairs, A with T and G with C. And RNA has bases also. And their bases like to pair up with DNA base pairs, but there's one difference. RNA has A, it has G, and it has C, but it does not have T. There is no T. And instead, it has U. And U stands for a base called uracil. And uracil, it's sort of like a distinguishing difference between RNA and DNA. Now, why is there uracil? I haven't gotten a good answer. I'm not sure scientists really know a good reason for this difference. But it does help with a lot of processes along the way. So I think it's just a way to tell the difference between RNA, because RNA's got a different job. That's my sort of opinion on it. And so RNA has to look at the actual message in DNA, and it has to rewrite it to this new code where there's no more T. And so it has to translate all of those T's, or transcribe rather, all of those T's into U's. So if you think of it this way, let's take a look at this. And so if you unzip that DNA, there we go. And then you have your RNA come in and transcribe. And so for every T, you're going to get an A. For every C, you get a G. For every G, you get a C. But then for every A, you're going to match it up with a U. So there's your first U right there in that message. There's your first U. And so now we have C to G, A to U, there's another U. And then T to A, G to C, A to U, there's a third U right there. And then C to G and G to C. And so now this RNA message, I'm going to translate that over here. So we have A, G, C, U, G, U, A, C, U, G, C. And so that's the RNA message right there. Now, your RNA will translate it will translate it like that. However, when you try to read that, there are words in RNA, and the words are very simple. They are only 3 letters apiece, these little words, and they're going to deliver these words to a part of the cell called the ribosome. We'll talk more about the ribosome later. But the ribosome is very simple. It can only read 3 letters at a time. And so you can break out your code here to 3 letters. So here's 3-letter sections. This one, incomplete. so there's only 3 complete codons in our little section that we figured out now. And so codons consist of 3 little bases in RNA. And so now, because it's one-sided like that, then that's why it's that single helix. It doesn't have another side. This doesn't stay here. The RNA will then kind of detach, and it's going to head for a ribosome. Now, how long did it take for me to describe that? Probably like 5-10 minutes. But now I'm going to show you a video that shows it in real time. And so we're going to see the processes happening in real time. And you'll see how fast this occurs. So I'm going to go, here we go, to my YouTube right here. I'm going to show you this little video. It is going to explain a few things along the way too. And so there's going to be a lot of proteins involved in this process, but let's take a look.
What you are about to see is DNA's most extraordinary secret, how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Okay. So let me come back. I'm going to unshare my screen. I'm going to come back to you. All right. So that process happens in, like I said, nanoseconds. And you have these big protein parts that come together. And then you have your RNA polymerase, which is the enzyme that helps to actually make that RNA, like pull all the pieces in for the RNA to be formed in that little string. And so essentially, it's as if that little RNA is going to match up with those pieces of DNA. Now we're going to make a little model that shows this. And so we're going to use the same parts as I did in DNA structure a couple weeks ago. And so we're going to need some tape. And so for this, I'm actually going to move my camera down a little bit so you get to see where I'm going to work, right? So you won't be able to see my face, but. And so now I have a couple things here. I have some pipe cleaners. That just gives your model stability. And so I also have some tape, and I have the popsicle sticks right here. We need 9 of them. So I'm just going to count that 9. Here we go. So there's my 9. Put that on the side. And I'm going to make my model. So now, first of all, the tape is going to be part of your backbone. And so here I'm going to use this as a reference. So I want my tape to be longer than that. So there we go. Not too much though. You want it to be pretty close to the size of the pipe cleaner. And now you want the sticky side facing up, but you need it to stick to the table. So this is where I just fold the edges down so the edges will stick and the sticky part will face up. So there we go. I'm gonna stick it down good. You use my thumbs to push down to make sure, yep, that would fit perfectly on there. So now I'm gonna get that out of the way. Now I need the other side. You don't want it to be too far apart. So you don't want it to be like over here. You wanna make sure that your popsicle stick will reach across. I like to have little ends overlapping, and you'll see it's really good for this RNA model that we're going to do. You'll see why that comes in handy. So now I'm going to do the same thing, about the same size. This guy out of the way. And I'm going to go about 2.5 inches. You could even use a ruler. 3 inches is fine. I like it a little closer. Okay, so that looks pretty good. Push down. Okay. So now I'm going to take my pipe cleaner, put it down the middle of the sticky side. There we go. And I'm going to use my finger, run it along the pipe cleaner so that the pipe cleaner sticks to the tape. Do that twice. There we go. Just try not to hit the sticky parts, then the whole thing comes off. It's always an issue. And so now same thing on this side. There we go. I run my finger along again. There we go. Okay. So now I'm going to take my 9 popsicle sticks. I like to cover up the edges. And because the pipe cleaners are like a nice thick border between the tape, you don't really have to worry about how they go down. You can always fix them later. So that's 2, 4, 5, 6, 7, 8, 9. And now I'm going to just straighten them out. There we go. Don't worry about your model being perfect. Yeah, it's just, this is for fun, more fun and learning. That's how that all goes together. Okay, so now looks pretty straight. Probably not perfect, but it's pretty straight. And so now I want to make sure they stay in place. Now, some people think that that's going to work. If you're good at that, go for it. But I am not. I will ruin my whole model. So I rip a nice long piece off and I hover it. And I'm going to kind of gently put it down. And then I'm going to squish it on. And with the pipe cleaners, you have to squish it so that you get in between. Otherwise, it won't stick together. Right, there we go. So squish it, squish it, squish it. If you're using a piece of wire in between, it's a little thinner, so you don't have to squish it as much. Now I'm going to do the other side. There we go. Again, gently try to line it up as best as I can. Looks good. All right, so now I've made essentially train tracks or a fence, whichever way you want to think of it. So it's not really much until you actually label it. And so in this case, like if you've done the DNA structure, this will look very familiar because this is what you make. But here we're going to do something different. Instead of this just being a simple DNA molecule, I'm going to make this side my DNA. So that side's going to be my DNA. And this side is going to be my RNA transcription. So RNA transcription. So I'm going to transcribe on this side what this DNA is. So I'm going to come up with any sequence I want for my DNA. And so, you know, I'm just going to randomly T, A, C, C, A, T, G, A, T, just any random sequence. Because you can look along your DNA and find any basic sequence of these little bases. And so, you know, let's give you your bases. Now, technically, if this were DNA, the whole thing were DNA, then you would match it up with its favorite pair. So like T likes to hang out with A likes to hang out with T, C likes to hang out with G, C with G, A with T, T with A, G with C, A with T, T with A. And so, but we're not doing that. this side is going to be your RNA transcription side. And so we're going to match it up with the matching RNA base. Now, it's basically the same rules, except A doesn't like T. There is no T in RNA. A likes U instead with RNA. Now, this is a T, so that doesn't affect us at all, because that means that that's going to go with an A on the RNA side. No, so that's no problem. Now you have an A. Oh, so there's your first U. So there's your first uracil, right there, your U. Now I'm going to keep going. So I have a C, that's going to be a G. I have a C, that's going to be a G. I have an A, that's going to be a U. And I have a T, that's going to be an A. And I'm going to keep going, G with C, A with a U, T with A. All right. So now I have, right here, I have my RNA transcribed. I have my RNA transcription from the DNA on this side. We would call that sort of like the template DNA, or so the original DNA that you get that copy from. And so now I'm going to put this over here so that we can see it a little bit better. And so now I'm going to adjust my camera a little bit. There we go. And so I have my DNA and I have my RNA right here. You can put little, if you want to put the hydrogen bonds in between, although they're not really connected for that long, because RNA is going to break off and bring that message outside of the nucleus. That's just how RNA works. And I'll show you that. We'll get to that soon. But first, let's take a look at our model. Now, remember, I said we have these words. This is fun. I have a little whiteboard here. And so right here, these three, so you have A, U, and G. That's my first RNA word. So that's my first codon. So that's codon number 1 right there. And then I have this one right here. That's my second codon. And then this is my third codon. Now I'm just going to rewrite these over here so we can see them better. So that one is A, U, and G. G, U, and A. C, U, A. So there's my 3 RNA words. And so those 3 RNA words are going to be taken to a ribosome. So over here, I'll just draw it over here so you can see if I have a cell and I have a nucleus. Here's my little nucleus. So I have all my DNA in here. I'll use a different color. You're going to get your RNA copy, and this is called messenger RNA, or mRNA for short. And so M for messenger, that down here, just so I can't think of it as secretary, but that's okay. Then that RNA is going to leave the nucleus, and it's going to find a ribosome. And so a little ribosome, I'm making it much bigger. Ribosomes are pretty tiny. I'm making it pretty big compared to the cell. But then that mRNA is going to bring the message to the ribosome. And the ribosome then is going to do the translating. So we go from transcribing to translating. Now, to show you that before we go any further, remember these codons, we'll come back to them. I'm going to share my screen again. There I am. Hi. And so I'm going to share my screen again, and I'm going to show you a quick video on how that works. Again, it's in real time. I love those videos that are in real time. So here we go. Okay, I'm going to go back to my YouTube. Get rid of this one. Oh, right. Stop. There we go. It's giving me an issue. And so let's go to my playlists, and I'm going to show you this.
The job of this mRNA...
I'll pause this and just make it nice and big so you can see it. Now rewind it. There we go.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides, or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
There we go. All right. Now, let me actually, I'm going to go back to here. All right. All right. So now let's go back here. So now you just saw how that RNA comes in and communicates with the ribosome. And so that ribosome, and they mentioned not only codons, so you have codons, but they mentioned anticodons. Now I'll tell you where those kind of come into play. And so they also mentioned transfer RNA as opposed to messenger RNA. So we'll talk about that. Now, to understand that a little bit better, let me go to another picture I've got right here. So now we're talking about how you make proteins. And so like how you make a protein called human growth hormone, how you make proteins that lead to your skin color and hair color. And so protein synthesis, which we call translation. So that's sort of how it works. It gets translated from the mRNA. And so now here's your mRNA, your messenger RNA, right there, and it comes in with the code. Now that code is going to have a complement. And so like, for example, if I use AUG, you're going to have UAC. That's going to be, that one would be the anticodon. It sounds like a bad thing, like, oh, the anticodon, like something that's bad, but it's not. And so now those anticodons are connected. So that's what we call the tRNA, the anticodon, the whole protein with that attached to it. And it connects to an amino acid. And so now amino acids, I like to call them the building blocks of proteins. And so I like to consider them the building blocks of proteins. We have a total of 20 different types of amino acids, although I hear there might be more now, but there's 20, kind of a stable number that we know about. We know how they work. And so those 20 amino acids are recycled within and used to make and build different proteins. And so the way they get built, so right here, we have a little section forming right here. We have a chain. And so the RNA will make a chain of amino acids. And then that amino acids, the way they are attracted to each other, when they come off, they're going to pop into different shapes. And so now, of course, well, you have that code. How do you know? Your body knows this instantly, but how do we know which anticodon or which codon is going to give you a certain type of amino acid? Well, the scientists have come with a chart. They've come up with this chart right here. Now, there's different forms of the chart. Sometimes they'll have a circular chart. Sometimes they'll have sort of like a block like this one. You'll see this in AP biology. So they have this chart in that book, just in different forms. And so it's sort of almost like almost looking like a multiplication type chart. And so, but if you have a codon, so now let's say that I have a codon AUG, because I've been talking enough about it, right? And I want to know what I want to know now. You don't have to worry so much about the anticodon. It's not going to really come into play. That's just how you get that particular amino acid. But I want to go from my codon and figure out which amino acid I'm going to get. And so I use this chart. Now, each one represents, each letter here represents a position. So you'll see the positions listed here. And so the A would be first position, U would be second position, and G would be third position. And so if I look at the first position here, I'm going to look down the column right there. And so I'm going to look down and find the A. And so here's my A right here. So that's my A. And so that means my amino acid is going to be located somewhere in this row. Now, I don't know which one. I don't know what box. I have no idea. I have to go to the second position to know that. So my second position is U. Now, here is second position right here. And this one we read across. So we read it across. So we're going to look for the U. The U is in the first box right there. And so if you think of it this way, I'm going to line this up like this. Okay, so now I'm not going to worry about anything in this box, or this box, because here is where they all come together. So that's where they all come together. Oops, get rid of that little line right there. There we go. And so that means it's one of these amino acids. Now we've got two possibilities here, and they're abbreviated. And so here's the abbreviations right down here. So one is ILE, and the other one is MET. And so if I look down, ILE, there's ILE, that's called isoleucine, sorry about that. And MET, I go a little more down there in alphabetical order, that's methionine. Okay, so I need to know which one. So I go to the third position, which is G. Now this one, you look in each section. This is the section we want, so I'm looking for the G in this section. Here's my G. And so if I carry that across, that means that I'm looking at methionine. So methionine is my amino acid that goes with that codeine. So that's how you use that chart. Now, if I take a look at I'm going to actually stop sharing my screen. I'm going to show you what we've got over here. Come back. Hey, there we are. Okay, so now I'm going to move this a little bit so we can see what I've got going on over here. And oh, here we go. And I have my decoder sheet. Now this decoder sheet is actually in the resources for this. So if you print out the RNA pages, you'll get this decoder sheet. You'll also get a couple of other helpful things to kind of help you remember how to do this. So I have my decoder sheet right here. And so now here are my codons. I made sure to put them on there. And so my first one, AUG. Now here's the funny thing, and this is sort of like where the AUG comes from. Scientists found out that at the beginning of every protein, there is methionine. So there's one little methionine amino acid at the beginning of every protein. We're not sure exactly why that is, but that's just the case. I don't know if there were any exceptions, but I'm going to say relatively that's usually the truth. And so right here, that happens to be mine. That was actually a total accident because I completely made this up. So that was a very fortunate accident right there. So there's my first codon, there's my second codon, and here's my third codon. So I want to know, okay, well, which amino acid is this going to be? And so I'm going to just get some markers. It's a little bit easier to see. And so we just did AUG. So actually we know that AUG is methionine. So here's my methionine. So I'm not going to mess up the whole thing for just that. But I am going to write this right here. M-E-T. Let's see if you can see that. That's really hard to see. So I'll do it in black. M-E-T. So that I know that AUG gives me methionine. And so there we go. It just happens that all your abbreviations are 3 letters. And so now I'm going to, I'll use this one. Now I'm going to figure out my next one, G-U-A. And so my first position is G. So right there. So I know it's going to be this row right here. My second position is U. So here's my U. So now it's going to be that column, except now we've narrowed it down right there to that box. So now here's the thing. I'm going to put this up a little bit closer so you can see it. Now you'll notice that in that box right here, it says Val, which by the way, Val is valine. So valine's right down here. And so that's what Val stands for. And there are no other choices. So technically, I don't need this last letter because I already know that's the only choice. But just to, know what I'm doing, if I look here, here's my A. So it would be this second to last one right here. That would be the right one, but that's valine. Now there's a reason for that. And so actually the way it works, if your RNA has any mistakes in the code, there you could still get, like if there's any mistakes in that third position, you can still get valine. You can get that no matter what letter is there. And so there's a few fail safes in here too. Now there's one I didn't talk about right here, the stop. That doesn't stand, that's not here. You'll see that there's no abbreviation for stop. That actually means stop. And what the stop is the end of a protein. So basically, that's where the protein ends. And so it just basically cuts off, and then there's your whole protein. And so that's where you might have stops. I have no idea if I have a stop on mine. I'm going to find out in a second. And so now let me just go back. So that's valine. So I'll use my red. V-A-L. Okay, so I've got 2 amino acids so far. I'll change colors. I'll go to green. And so now my last one, C-U-A. And so my first position is C. And so now I know that's going to be my row right there. U, lots of U's in the middle. That's a coincidence. And so now I see again, I have one with no other choices. So this is going to be L-E-U for that one. And so this one's L-E-U. And if I go down here to L-E-U, that is leucine right there. So leucine. And, but just so I get used to doing this, U. So if I look here, the U is the first one. So it's this first position. And again, that last letter really doesn't matter. So there's a lot of different ways that you can get leucine. And so you can see that happens a lot. So there's a lot of ways that, you can make a mistake, or, these are important. It's important that you make a lot of this. So, I don't really know exactly why there's a lot of repeats, but it does make sense. And so it makes sense. Whereas if you're up here, then sort of you have a 50-50 shot. I'll bring that up a little bit closer. So you have a 50-50 shot of getting a stop or a TYR, which is tyrosine. I just happen to not have a stop in my particular model. Now, what's fun is that you can actually do this and make your models. You can make them longer, and you can create like little amino acid chains and see what you get. And so you have your little ribosome, and you make your chains. The chains will always start, well, they're supposed to start with MET, but when you make your own, you might not have a start like that. And then you can fill it in. Like, for example, I have valine right here. I have leucine. Oops, there we go. LEU. And then you can fill in your amino acids and see how that works out for making a protein. All right, so now that's how RNA transcription and RNA translation work. I hope you have a lot of fun working with this. I find this to be kind of a fun thing to work with, to figure out amino acids and how they all come together. And I hope you enjoyed it. If you have any questions, you can put them in the chat even after it's done, like after the chat's over, you can put them in the comments below, and we will keep an eye on them and answer what we can. So I hope you have a great afternoon, and I hope all is well in quarantine land at the moment. Have a great day.